much, the organizers, the audience, for having me here. And it's a pleasure to present what we are doing as neuropathologists, using also computational methods in our daily routine. I think I have to click here now, share. Do you see the presentation?
Yes, we see the presentation.
Yes, okay. So, Methylome, we have heard of it a bit, is a layer, and I would say it's a novel, maybe not so novel anymore, but it's a layer in diagnostic pathology that, at least for neuropathology, has now become standard repertoire. And in fact, if you look at neuropathology or pathology in general, you have these layers. So you have a clinical layer that can be complemented by macroscopy or an imaging layer. So you have, for instance, a process that is somehow destructive to the brain. And then let's say you took a biopsy from this lesion, you would see a histological image, which we have heard about today quite a bit, that contains patterns, that contains some information. Then below that, I would put the methylation layer here, depicted by this microarray picture, because you will see that's still the standard method to detect methylation level on a genome scale. And then below that, you could also have chromosomal levels like copy number alterations. And then even below that is what we have heard a lot about today. That's the sequence level, the DNA and RNA sequence level. So in fact, methylation analysis, in the way neuropathology currently does it, is more like a form of molecular histology. So it's kind of more related to these ones than to these ones, as you will see in the following. And this is a title slide from a magazine that I found some time ago, which I think is pretty characteristic of the epigenome. So here you have the DNA, and on top is something that looks like switches, like binary switches, and hence I'd call the methylome, that's kind of the bits of cancer. Well, what are those methylome bits useful for? For one, it has been known for a long time that, for instance, one could analyze MGMT promoter methylation state as a correlate for MGMT expression, which is a DNA repair enzyme. And if a cancer doesn't express high levels of DNA repair systems, you could have a higher chance of a therapy response with alkylating agents, for instance, that cause DNA damage that would get repaired by that enzyme. Or you could look at MLH1 promoter methylation, which could be prognostic in certain types of cancers. Nevertheless, all you need is the methylation information to get to this. And luckily, there is a monopoly. There is still currently a monopoly on methylation detection on a genome-wide scale, and that is a microarray. It's essentially a modified SNP array developed by Illumina, which detects methylation on a set of currently 850,000 genomic sites. And due to that monopoly, we have collected, or the world actually has collected, a huge number of methylation data that all are in the same shape. So essentially, it's a tabular format, like from any microarray. So you get a value between 1 and 0 for the methylation of each of the represented CpG islands, which means it's perfect for machine learning applications and to curate a sample database, because the methodology doesn't really impact so much on interpretation as it does for other techniques. If we had only methylation information, then we could probably find tumor types, because methylation, as I will show you in the next slides, is kind of a tumor type surrogate. A sequence, a genomic sequence, doesn't define a tumor type alone. A methylome, on the other side, defines a cell type. A cell type because each cell type in the body of a multicellular organisms, but also in more simple life forms, defines how a cell works. And if we had a sequence and the methylome, then we would probably know the tumor type and we could describe it, but it would be a very tedious procedure to do so on a routine basis. And hence, if we only had a methylome, actually only had a glimpse at the methylome, because the methylation array is only a glimpse at the methylome, it's not a full genome methylome, then we have a pretty good tumor type surrogate. The standardized data acquisition is already solved due to this Illumina monopoly, which started out many years ago. And hence we have created a rather large database of cases that could hopefully be an infinite repository of cases. It allows us to determine novel entities that are not recognizable on the microscope, or that are not recognizable on the microscope due to their rarity of occurrence, because no single pathologist would ever see two in his lifetime. And it could likewise, as with the other parameters that we have heard about today, be a predictor of biological and clinical behavior. So what can the methylation bits do and decide? I like to make this comparison. And that's a Dipswitch. A Dipswitch, as you probably know, in older computers, served to select programs that should be running on that same machine. Let's say that machine is a cell. It has a chip in there with all the genetic information or all the program code. But depending on how this switch is being set, different versions of the program would be executed, and it would either behave like a game or like a typewriter. And pretty much the same on a much more complex level occurs with DNA methylation as one of the epigenetic modifiers that is very well studied. So you have essentially a binary system containing zeros and ones, and these set of zeros and ones ultimately determine tissue differentiation, even though all these cells share the same genome. The same, of course, occurs in tumor cells, and a lot of their lineage is preserved. So how do we detect DNA methylation in clinical routine? And the key question there, the key clinical question is, how and when do we detect these? So we could do DNA bisulfite conversion that converts methylated cytosine differently than unmethylated cytosine. So unmethylated cytosines gets changed in the end to uracil and then to thymine in a DNA molecule, and methylated cytosine is protected from this modification. Hence you generate SNPs, and these SNPs could then be detected by any common technique like PCR, qPCR sequencing, or SNP arrays, as is the Illumina B-chip array. Or you could also have MLPA, for instance, with methylation-specific restriction endonucleases. On the other side, then we are down to hours. You could do direct epigenetic sequencing of native DNA with nanopore sequencing devices. Once you had such methylation files that describe the tumor, then it's like looking at images and doing image comparison or image reverse search, as you probably all know. So you can feed in an image, ask a database, what is that image similar to? And you would get some images that have already been annotated, and you would have your diagnosis or your tumor type. In just the same way, it's what we do with our methylation files. We ask the AI if the AI had ever seen that pattern or not. And this has been prominently developed and led by the German Cancer Research Center in Heidelberg. And the neuropathologists there started a while ago to collect methylation data sets and found something astonishing. So what you see here is a TSNE plot of methylation data sets from the Infinium B-chip array. and they have been colored with clinical diagnosis, so with clinical annotations. And the TSNE, of course, knew nothing but the methylation of these, the genome-wide methylation patterns. These are 2,801 brain tumor samples. And you see that these colors, they suddenly start to group nicely, even though nobody had told the system what these entities were. So this is an example of unsupervised machine learning that somehow detects the same as the observer would have detected. However, if you look now at the pathological diagnosis of cases in a validation cohort, and we were also part of this validation cohort, where we checked what was the histological or genetic or sequencing diagnosis for brain tumors, then you see instead of straight lines, you have a lot of diagnosis changes here. And the horrible thing for us neuropathologists, because we had a pretty high confidence in ourselves, was that there were many, many diagnoses wrong, like 12% of misdiagnoses were across expert centers in neuropathology, and almost 3/4 of them were WHO grade changes. And this means a different treatment in clinical terms. So every 10th patient approximately would go out with a wrong diagnosis. And all that led to the acceptance of this methylation-based classification of brain tumors. I'm going to show you a clinical example of such an application. Look at this radiograph. Here you see a lesion, and this lesion, all the neuroradiologists would agree, this lesion doesn't really show high-grade malignancy. Also, histology alone doesn't really tell you much. Of course, if you would have sequenced it in depth, you might have found something, but on a standard sequencing approach at that time, you would only sequence IDH and you would not find any mutation. So you would say that's a diffuse astrocytoma and it's kind of benign. However, the classifier, if you took the methylation data set from this file, would tell you this is a glioblastoma, meaning the most malignant brain tumor of the adult, also the most common form. And this diagnosis in terms of histology would be completely incompatible with this image. However, if you looked at the genetics behind this, and here you could derive a copy number plot from the methylation data, you would see a classical glioblastoma signature. You would see a gain of chromosome 7, a loss of chromosome 10, an EGFR amplification, and a P16 deletion. All these are findings. If you show them nowadays to an oncologist, they would say, this is glioblastoma IDH wild type, no question about that. But you also see this on the methylation profile. And this is what happened to the patient at the time. One didn't really intervene. This was really in the beginning of methylation diagnostics. And one said, well, we don't know what to do here clinically. So we wait, and this is what happened. So the full-blown picture of a glioblastoma is there. And we have seen by now many of these cases. And now things start changing, and oncologists start treating these patients in advance. Another application for such profiling is meningoma. It's a, for most cases, benign tumor on the coverings of the brain. However, there are 15 histological variants, and there are 3 WHO grades. And grade 1 would be benign, and grade 3 would be malignant, and grade 2 would be somewhere in between. And if you look at this, here you have a histological stratification of the progression-free survival of these tumors with the 3 histological grades. In fact, it turned out that there are about 6 or maybe even more molecular types of meningoma that are very much unlike each other, despite their microscopic appearance. And 3 of them follow a rather benign course, 2 an intermediate course, and 1 a malignant course. And you see a much higher stratification of clinical behavior based on the methylation pattern compared to histology. As of now, in Basel, we have in our daily routine analyzed over 3,000 tumor methylomes. Of course, we also get external samples sent to us. And we started seeing cases that are unclassifiable by current classification systems, such as the published brain tumor classification in Heidelberg. So we thought it would be nice, at least for these cases where we don't have enough backbone data, to build up a database that would be able to subject them to unsupervised machine learning. So we could at least look which cases in our database are similar to those cases, to at least have some clinical information for every case that is submitted for analysis. And therefore we developed Epidip. It's a platform that uses standard dimension reduction. Here we use uniform manifold approximation projection, simply because the algorithm is much less resource consuming than classical TSNE, for instance, and it has been ported to GPU processors. So within minutes, we can actually run a case through that system against currently 18,000 reference cases that we collected from various repositories like Gene Expression Omnibus and the TCGA. And there you can now classify against whatever cancer has been deposited as IDAT files, as methylation files. For instance, here is a breast cancer cloud, and you could see where your particular case localizes. Everybody's allowed to upload anonymously cases to this system and check where they actually fall. So it could augment routine pathological diagnostics. You can also use it as a discovery tool. Of course, you can zoom into that plot. Here you see quite an astonishing example of methylation signature relevance. So these are both melanomas. These are melanomas and melanocytomas of the uvea, so CNS-derived melanocytes that go malignant at one point. Whereas here in the upper cloud, you see cutaneous-derived melanomas, and they are obviously very different, even though under the microscope, these look pretty much the same. Now, I'm going to switch gears a bit, because now we were talking about microarray analysis, and microarray analysis, as you probably know, is a slow process. And this process is too slow for clinical practice. In fact, it's much slower than histology. It's much slower than immunohistochemistry. It's even slower than sequencing and FISH. So a neurologist from Berlin, Philipp Euskirchen, he's actually an impatient guy. And he said, I need the results faster. I want the same kind of machine learning approach. I want a classifier, for instance, a random forest classifier. I need a copy number profile that I have an independent look at my data, and I want it fast. And what he did is he transformed the whole idea to nanopore sequencing. Now, nanopore sequencing is a technique that directly sequences not only the DNA base sequence, but also the base substitutions on native DNA. And in the end, you can at the moment type brain tumors and the meningomas here for the grading, and you have your molecular profile in less than 24 hours. In fact, it takes about 4 hours, including the microscopical examination on a frozen section, to get these data. And of course, we are aiming to go in the below 1 hour, which I think is feasible. So how does this work? Nanopore sequencing is dragging a DNA strand, typically a single strand of DNA, through a proteinaceous pore molecule, and looking at the ionic current that can still pass besides the molecule. So it's in fact a molecular Coulter counter or hematocytometer-like device that works with many parallel channels. And from each of these channels, at a rate of approximately 400 bases per second, you can get this electrical current that, again, by a neural network, is then translated into bases or epigenomics, barcodes, etc. And with those reads, You could then do a genomic alignment, do copy number counts, and then you could read the methylation, etc. And then you normalize your data, you plot it, and you can apply dimension reduction, you can apply random forest classification or whatever machine learning. And even though that's just a glimpse of the data you would get in an array, still that data density is sufficient to make a proper diagnosis. Here's a benchmarking that we did before we introduced it into our routine diagnostics in 2019. So we typically work with intraoperative frozen sections. This does not work on FFPE, but we don't need FFPE anymore for brain tumors anyway. So here we take a couple of frozen sections. We extract the DNA with a commercial kit, and then we subject that DNA to direct sequencing on these devices. If you use a Minion device for a typical brain tumor, 1 hour of sequencing is sufficient. It can even go down to half an hour if you increase the amount of DNA. And you can do live data analysis with NanoDX, that's the algorithm pipeline, during the run. And I'm going to show you how this looks like. So pay attention to this one here. That's the reads coming in, the reads of native DNA. These have approximately 1,000 base pairs in length. Here you see a copy number profile arising, and here you see the classification error of that random forest. So in the beginning, when the run starts out, your cross here, that's the case that you're looking at here, will probably jump a bit, but then you will see as the more data pops in, this TSNE plot here will be resolved. And you start seeing a signature here also on the copy number profile. You start seeing here a dominant classification by the random forest classifier. And you see in that green cloud, the cross is always in the green cloud. So our case is always in the green cloud. And the more data we acquire, the better the copy number profile gets resolved, the tighter the clouds get here of the reference samples. And that's the same reference samples that are used in the German Cancer Research Brain Tumor Classifier Center. And in the end, you end up with a diagnosis with a copy number plot, chromosome 7, EGFR amplification, P16 deletion, loss of heterozygosity of chromosome 10, and here that green cloud would be glioblastoma IDH wild type RTK1, as it is also depicted independently by the random forest classifier. And here that's the microarray. In the end, of course, we run all the cases through microarray as well, and you see it's pretty much the same thing, but with the difference you get it in approximately 2 to 4 hours. The price, for instance, for this is approximately 200 Swiss francs, including the sample preparation, the DNA extraction kit, and all the materials that you need for the whole thing. So far, we have over 200 brain tumor cases analyzed. Data accuracy is really high. We get approximately 1 kilobase read length and typically between 1,000 and 6,000 CPG sites that overlap with the array data, so with our ground truth data. The misclassification error is really small and pretty much never clinically relevant. The CNVs are still a bit of a problem because of data density. So focal gains, focal losses, if they're really short, that can be missed by this technique. But as a first glance methodology, it performs much better than, for instance, histology. It was also benchmarked, the method, in a multi-center approach. Now an ongoing European-wide validation study has been initiated by Philipp Euskirchen. And so far, we don't really see discrepancy. So we have already integrated as an in-house validated diagnostic test in our repertoire. This is how the routine looks like. So the neurosurgeons give a intraoperative frozen section and a definitive biopsy. This one is then taken within minutes to the microscope, is checked whether the tumor cell content is okay. The neurosurgeons actually ask us, is the material enough for nanopore sequencing, or do you need more? And then within a couple of hours, usually by the end of the day, we render them a complete molecular diagnosis of that brain tumor and integrate it with the WHO classification. So where do things go from here? What I think is, and we have seen similar slides today quite a bit, is that this is an endless circle. It's an endless circle where you always start to feed in data, and you try to feed in, of course, in a very standardized manner, because that's key, that the data and the annotation and also the clinical outcome data get recorded and get available, so that the system can be enhanced just by the simple number of data points. The other thing where things might go is adaptive methylation sequencing. Some of you might have heard that you can actively take influence on the Minion sequencer. That's actually a Minion sequencer. It's a USB stick-sized device. And you could then reject reads that you no longer need. So to enrich for those DNA sequences that are of higher interest. And since all this platform runs in live mode during the sequencing, you could use that to decrease sequencing time and increase precision. And the other thing we are working on is to port the entire platform away from a PC server that consumes a lot of resources and power down to a GPU-equipped embedded system, so that the entire platform could really be portable and could really fit into every lab at very little cost, so that this one could eventually move into the frozen section room and enable all pathologists to use this method. And it would actually be cheaper than producing a histological slide. With that, I'd like to come to the acknowledgements. I'd like to thank our colleagues from neurosurgery who are really open for new methods, also computational methods, and support us in every sense. Then to Life and Brain, who is doing the array analyses for us. To the people from the German Cancer Research Center. David Kappe is now at Charité, working together also with Philipp Euskirchen. Then to the people from our R&D team from the MolPad labs, then to the Oxford Nanopore support team, who give us a lot of deep insight into their platform, so we could develop our tools, and to Miles, who is also a user of ONT and who helped with the integration on the NVIDIA platform. With that, I'd like to thank you all for your attention. And if you have questions, please feel free.
Well, thank you so much for your very interesting talk. I'm always fascinated by nanopore sequencing and its promises. I think we're actually just about at the, we should actually move on into the break, but I think we have time for a couple of questions. I have just a quick question. Do you think pathologists will go into kind of this notion of just doing sequencing to make a diagnosis without having a histological or visual proof of the lead? I mean, that's what you mentioned at the very end. I'm intrigued by that, obviously, but are you convinced that this is a direction where we can go?
I think maybe I made my point wrongly. Of course, we are looking at the slide, at an H&E slide, to make sure that we are actually sequencing tumor, but we are only looking at a frozen section. And for many brain tumors, we skip immunohistochemistry or looking at the paraffin embedded slides, simply because we already have the molecular profile before the FFPE section is there.
Very interesting. Thank you.
But we should always look at the morphology to know we have tumor in there.
Good. Victor, you had a quick question. I was not sure. No, you're good. Okay, I think the hands are all. Abdullah, you have one. You have a comment.
Yeah, I mean, we don't have much time, but I just quickly wanted to ask, Jürgen, so for brain tumors, it's also very often difficult to extract biopsies. So would your tool, I mean, the methylome detection also work with liquid biopsies? So, you know, when you take blood, basically, is this something that one could also consider?
Yeah, there have been many reports on this to detect methylation, aberrant methylation in liquid biopsies, but by neither of the techniques that I showed here. So here you would need an amplification-based technique that would pick out specific sites from the DNA, select for specific, so it would be a targeted sequencing approach in the end, or another capture approach, and then use the methylation sites that are really aberrant from any other methylation pattern in the entire body in any cell. And then you could detect, for instance, there have been reports on breast cancer, ovarian cancer, lung cancer, and so on, that you could predict that a person is developing these cancers based on aberrant methylation patterns on the cell-free DNA in the blood. But this would not be by the array, and it would definitively not be by nanopore.
Okay, thanks a lot. Great, cool. Thanks, Rido.